See, the subject of linkage and crossing over has always been one of the important aspects in the field of genetics because this gives us the idea about the location of genes on the chromosome. And when we study about crossing over, that gives us the concept that how crossing over produces new combinations of genes, and such new gene combinations make raw material for the evolution to act upon. If we see this phenomenon, actually we find that on a single chromosome, a number of genes are located. Since every individual possesses hundreds and hundreds of genes, and those genes will have to be situated on just few chromosomes, so it is quite possible that a single chromosome will harbor a number of genes. So they will be definitely linked to each other. So since the genes are linked on the chromosome, the phenomenon is referred as linkage. But there will be substantial area or gap between the two genes, and in that area there will be crossing over. So as a result of such crossing over, new combinations will be formed. And see here, during prophase division, during prophase 1 of meiosis, there is a substage packaging. So during packaging, the crossing over takes place. And we know that by that time, each chromosome, suppose this is a chromosome and this chromosome has duplicated. Now it is being represented in the form of its sister chromatids. So these are two sister chromatids of a particular chromosome, and this is its homologous chromosome. So A and A1 are two homologous chromosomes, and each one is represented by its sister chromatids. So crossing over occurs between the non-sister chromatids, means one of the chromatids of A and the one of the chromatids of A1. So crossing over will be occurring between non-sister chromatids of homologous chromosomes. These two are homologous chromosomes, and as I just referred, that by this process new combinations of genes will be formed, and such new gene combinations will form a raw material for the evolutionary events. Now this aspect we can understand by taking two cases, one 2-point test cross and the other 3-point test cross. When we are studying crossing over between two genes like A and B, then since these are two genes, so two genes means 2 point, then that will be considered as 2-point test cross or 2-point crossing over. And when we study crossing over between three genes like A, B and C, then that will be referred as 3-point test cross. So this aspect I will explain in case of Drosophila melanogaster, because this is a species in which hundreds and hundreds of mutants have been recorded. They are being cultured, and there are laboratories where such mutants are being reared, so they can be used to study this phenomenon of crossing over. And let us consider the first one, that is 2-point test cross. So to study this aspect, we need two types of flies, the wild type flies and mutant flies. Suppose you have wild type flies which in their phenotype are gray bodied and normal winged. So they will have actually normal body color as well as normal wing structure. So they are normal flies, and you can take females of such type, that is normal types, and you can cross them with the mutant fly. So mutant fly means that will have black body color and vestigial wing. We know that these two alleles, that is alleles deciding for black body color and vestigial wing, are situated on the second chromosome. So you can take a mutant stock having black body color and vestigial wing, and they could be crossed with each other. In next generation, you will find normal phenotype in the flies, and then such flies will be crossed with the mutant one, and that will be referred as test cross. So let us take this particular example, but here I am simply making you acquainted with the different symbols. B plus stands for gray body color. This small b, that stands for black body color. So this Vg plus, that represents normal wing structure, and Vg represents the vestigial wing structure. Now here is the cross. The flies which were selected for making initial crosses could be wild type. As I told, they are gray bodied and normal winged. They will be crossed with flies which are mutant. They are male flies, and they have black body color and vestigial wing. If you cross them in the next generation, you will find all flies having the gray body color, that is normal body color, and a normal wing structure. But in these F1 flies, you will be selecting only females. You will have to consider, you have to actually sort out virgin females to study crossing over between these two genes, because in Drosophila melanogaster it is well established and documented fact that crossing over occurs only in females. So you will have to select F1 females, and these females will be crossed with the mutant males which are having black body color and vestigial wing. So this kind of cross will be referred as test cross, because we are crossing the F1 flies with the mutant flies. So in the next generation, you will find 4 different kinds of flies. That is, flies with normal phenotypes. There will be males and females having normal phenotype, that is normal body color and normal wing structure. Then you will have flies which will be totally mutant, means males and females flies will be there, which will have a mutant, a black body color and vestigial wing. So these flies will be there, and you can call them as parental flies because you started with such kind of parents. So these are referred as parental flies. They show their phenotype exactly similar to their parents. But you will have 2 other kinds of flies which will be normal for one phenotype and mutation for other phenotype. Like you will have gray bodied flies but having vestigial wing, and also you will have flies with black body color and normal wing. So these flies, they are actually recombinant flies. They are obtained only because of crossing over between the two genes. See here in this structure, if this exactly this chromosome comes in the gamete, then it will be B plus and Vg plus, and this gamete will be combining with the gamete coming from the mutant fly, that is with B and Vg. So such fly will have normal phenotype, it will be of parental type. Likewise, if this chromosome B and Vg combines with the gamete coming from the mutant male also with BVG, then this will be a fly with black body color and vestigial wing. So these 2 types will be parental types, as I just showed in the class. But you may also find individuals in which there will be a gamete with B plus, and that will combine with VG, and that chromosome means that gamete in which B plus and Vg that combines with B and Vg, then such individuals will be gray bodied but vestigial winged. And the 4th situation will be that if crossing over occurs here, the other recombinant type will be B and Vg plus. So that will be combining with B and Vg, and you will have flies with black body color and long wing. So because of this reason you will find, so these two are recombinant types. Now we can know that what is the difference between these two genes, that is B and VG, and that we can know by calculating crossing over. So what we will be doing, that this crossing over, percentage of crossing over, that can be known by considering total number of recombinant flies. All those flies which are recombinants, their number will be considered, and that number will be divided by total number of progeny observed, and the product will be multiplied with 100. So this will give us the idea about crossing over between B and VG alleles. So see here, there is a data. In this case, you can consider that F1 female fly was actually crossed with mutant, and you got these 4 different types. That is flies having B plus and VG plus, these are parental, and BVG, these are parental. They are 830 and 800 in number. Then you have recombinant types like B plus and VG, those with gray body color, and then vestigial wing, they are 118 number. Then you have flies with black body color and long wing, they are 119 number. So you have to simply count the number of recombinant types. So if you count 180 plus 190, then that comes, that number will be here 180 plus 190, and this number will be divided by 2000. So total of recombinant types is being divided by total number of progeny observed, and that gives the value 18.5, and that comes in percentage because you are multiplying it with 100. So you get 18.5%, and this 18.5% is crossing over occurring between B and VG genes. So the distance between these two genes will be 18.5 centimorgan. Centimorgan is the unit of crossing over. So it means the distance between these two genes B and VG is 18.5 centimorgan. Now let us see 3-point test cross. Example is again Drosophila melanogaster. As I told in the beginning, so many mutants are there in this fly, and that is why we can see that crossing over occurs between 3 genes, and what is the percentage of occurrence of crossing over among them. So now in this 3-point test cross, as I told in the beginning, we need actually 3 different types of mutations in the same individual. So you may have flies having sepia eye color, which is represented by SC, then flies with curled wing, that is being shown here by CU, small cu, and ebony body color, that is similar to black body color but it is lighter black, and that is indicated by small e. So see, genotype wise, those flies whose genotype will be SC, CU and E, and SC, CU and E means on both homologous chromosome exactly the recessive alleles are there, then such fly will be showing sepia eye color, curled wing and ebony body color. But if these 3 alleles are normal alleles, that is SC plus, CU plus and E plus, and on the other chromosome also SC plus, CU plus and E plus, then such flies will show all normal features, that they will be red-eyed instead of sepia eye, they will be normal winged instead of having curled wing, and gray body color instead of ebony body color. So they will be normal in their phenotype. So you need these 2 stocks, that is the wild type stock, and from this stock you can collect the female flies. So virgin female flies will have to be collected from the wild type stock, and these flies will be crossed with mutant stock. Okay, mutant stock. So what will happen in the next generation? You will have heterozygotes, and male and female heterozygotes will be there, in which one chromosome will be carrying SC plus, CU plus and E plus, and the other chromosome will be carrying SC, CU and E. So this situation will be there, which you can see in the next slide. See here, you actually crossed the normal female flies with the mutant one, and in F1 generation you got flies having SC plus, CU plus and E plus, and SC, CU and E. These are heterozygous flies, but they will be crossed with the mutant fly, that is having all the three mutations on both chromosome, on both homologous chromosome. But one thing which I have not shown here, but these heterozygous flies will always be female. So these will be female flies. Keep in mind that heterozygous flies which you will be using for crossing over, for studying this phenomenon, will be female flies, because in Drosophila female you can see crossing over, and these flies will be test crossed with the male flies, which will be mutant one for these three genes SC, CU and E. So when you will cross them, you will find 8 different types of flies. But before that, let us see about the gametogenesis. So if gametogenesis will occur in the mutant one, then all the gametes formed will have the chromosome on which SC, CU and E will be present, because even if crossing over occurs between these genes, there will not be any effect. So all gametes coming from this male individual, which is recessive one, will contribute a chromosome with SC, CU and E. But in case of female, you will find 8 different kinds of gametes. So this is important to understand how 8 different kinds of gametes will be formed. That I can explain here itself. See here, one chromosome on the upper side. This chromosome as such may go in the gamete. So SC plus, CU plus and E plus, it may be going in the gamete as such. Or the other situation will be SC, CU and E. This chromosome will be going in the gamete. Okay, so you can say that these are the chromosomes which have not experienced crossing over, and they are going in the gamete. If crossing over occurs between SC and CU genes, if between these two genes crossing over occurs, then you will have 2 gametes. So I have shown them side by side here. On the upper side you can see these 2 gametes will be formed because of crossing over occurring between SC and CU. See how this will happen. If crossing over occurs here between SC and CU, then it will be SC plus and CUE. One gamete will have this chromosome SC plus, and then because of crossing over, the rest of the portion from this chromosome will come. So it will be CU and E. So SC plus and CUE, and the other will be SC and the upper portion, that is SC included with CU plus and E. So CU plus and E. So these 2 gametes will be formed due to crossing over between SC and CU. Now if crossing over occurs between CU and E, then what will happen? You will have again 2 types of gametes, and that is shown here in the lower side. So crossing over is now occurring between CU and E. So you will have SC plus, CU plus and then E. SC plus, CU plus and E. And the other situation will be you will have SC, CU and E plus. So SC, CU and E plus. So these 2 types of gametes will be formed when there will be crossing over between SC and CU genes. Now you have seen what would be the result of crossing over if occurs between SC and CU, and also between CU and E. But there will be another situation, and that will be double crossing over. That is, crossing over may occur between SC and CU, that is SC and CU, and then again it may occur between CU and E. So if crossing over occurs at both places, then what will happen? You will find gametes containing SC plus, okay, SC plus Cu and then E plus. So SC plus Cu and E plus. And the other situation will be SC and Cu plus and E. So SC, Cu plus and E. So this way 8 different kinds of gametes will be formed in this heterozygous female. This thing you have to understand. During meiotic division, prophase 1, Paquetin stage, this will be the event taking place. So that is the stage, during prophase 1, there is a sub-stage, that is Paquetin stage, when crossing over will be occurring between non-sister chromatids of homologous chromosomes. I have shown here only 2 chromosomes, one with normal alleles, other with mutant alleles, and the result of crossing over will produce 8 different kinds of gametes. What will happen? These gametes will be fertilized by the gamete coming from the mutant male. So with all these 8 gametes, you can simply add up this one, SC, Cu and E. SC, Cu and E will be added with everyone. So what we will find, that even if it is being added with the first one, you will have a fly with normal features. If it gets added with the second one, you will have flies with all mutant features. So these will be parental types. And then you will have recombinant types. So let us see how these can be explained. In this table, the first 2 cases are of parental types. See, this is the parental one with normal features. This one is a parental fly with recessive features. This will be a fly having the normal eye color, because SC plus stands for normal eye color, and then curled wing and ebony body color. Then here you will have fly with sepia eye, then other 2 features, that is wing will be normal and the body color will be normal. So that will be obtained because of crossing over in the first region or between SC and Cu. Then you will have those flies which will occur due to crossing over occurring in the second region, and then you will have the double crossovers. Now if you perform exactly the same experiment and observe the F2 results, then this number you may be getting. Exactly, suppose these are different types. Out of them, the initial these 2 are parental types. Then those flies which occur due to crossing over in the first region, that is SC and Cu. Then these are the flies which occur due to crossing over in the second region, that is Cu and E. And then these 2 types are the flies which result because of double crossing over. Now you add up their number, it comes 2000. So what we will be doing, we will see what is the crossing over percentage if it occurs in the first region, and then if it occurs in the second region. So here again the same formula will be used, that is number of recombinant types divided by total number of flies or individuals observed into 100. So let us say crossing over that occurs in the first region, that is between SC and Cu genes. What we are doing, we are simply adding the number 230 plus 220, and along with these you have to add the double crossovers. So double crossovers, they are 16 and 14 in number. So this number 16 and 14 you have taken here. The total number comes 480. Okay, and this 480 will be divided by 2000. It comes 0.24 multiplied by 100, because you have to find out percentage. So it comes 24%. It means there is a distance of 24 centimargan between SC and CU. These two genes are located 24 map unit or 24 centimargan apart. Then you can consider the second reason, that is crossing over occurring between CU and E. What you will be doing here, you will add up these numbers, that is 190 plus 180, which are being obtained by crossing over in the second region, plus the double crossovers. So I have done here 190 plus 180 plus 16 plus 14, because double crossovers will be added in both the cases. So that gives the number 400. 400 divided by 2000, it comes 0.20, and multiplied by 10, 20. It means 20 map unit or centimargan distance is there between CU and E genes. So this is the way you can calculate crossing over between the genes, and you can know the distance, what amount of distance exists between the three genes. Now you can map these three genes, SC, CU and E. See, this is a chromosome. We know that these genes are located on the third chromosome, that I will show you just now. In Drosophila, there is a third chromosome, autosome, and so many genes are located on this chromosome. Out of them, we have considered just three genes, and a gene which causes sepia eye color and curled wing and ebony body color. So SC and CU. Okay, this one is CU. So the distance between this, that has come 24 centimargan, and that you can show here. Then between CU and E, crossing over percentage came 20, and that is why this distance is 20 centimargan. So these are the steps you can apply, and you can know the distance between the adjacent genes. There is one more phenomenon, and that is interference. That is how much interference occurs if crossing over takes place in one region, then whether it interferes with the same occurrence, with the occurrence of crossing over in the adjacent region. So for this calculation, there is a method, and that is once we calculate coefficient of coincidence. This coefficient of coincidence has to be calculated, which is shown by letter C. This C is equal to frequency of observed double crossovers divided by frequency of expected double crossovers. So what we have observed? We have observed that the total number of observed cases, that is double crossovers, you can see in this case are 16 and 14. So they are actually 13 number. So this number we are taking 30, and this 30 out of 2000. So that will become frequency of observed double crossovers. We are observing 30 double crossovers. So 30 divided by 2000, this value will come 0.015. And then we can also calculate expected this frequency, and expected frequency can also be calculated from the obtained values, like this frequency has come 0.24 in the first case and 0.20 in the second region. So 0.24 into 0.20 means we have to take the frequency actually, not the percentage. So it has come 0.24 for the first region and 0.20 for the second region. So we will multiply these two values, and when we multiply this, the value comes 0.048. So that becomes the expected double crossovers. So you will be dividing 0.015 by 0.048, and that C value comes 0.31. It means the coefficient of coincidence is 0.31. Then interference you can calculate, because I is equal to 1 minus C, where C value you have just obtained, it has come 0.31. So this 1 minus 0.31, the value comes 0.69. It means there is Positive interference, yes, we can say that interference is there, because this interference could be 0, this could be means 1, this could be even in negative value, but here we find that positive interference exists, and there is 69% interference in the crossing over. It means that if crossing over is occurring at one spot or between two genes like SC and CU, then that is interfering crossing over at the other region. Means double crossover, since they are not so many in number, it is only because of interference, and that interference is of 69%. So, and here I have shown the map, means by following such crossing over attempts, people have mapped genes, mutant genes on the different chromosomes in Drosophila. See, this is X chromosome, which is referred as first chromosome. This one is second chromosome. This one is third chromosome. And this is fourth chromosome. Fourth chromosome is very tiny, and that is why only few genes are located on it. So you can see the number of genes located and their sequence on the fourth chromosome. Here we selected genes of the third chromosome. So sepia eye color gene is located here, and then you have the other one as curled wing. So definitely the curled wing gene is located here, and between you can see so many genes are there. And the third one is for ebony body color, it is also located here, and you can see the distance between them, that is shown on this left side. So these are the things you can go through this map. It is for Drosophila melanogaster. First chromosome, that is sex chromosome, the genes located on it are shown, and such genes could be used to study X-linked inheritance pattern. So this aspect is quite important, and once again I will say that crossing over is one of the important phenomena that produces new combination of genes.